This video explains the topic of genetics, especially with regard to Mendel's work. So Gregor Mendel was a monk who lived in the mid-1800s. He began the process of modern genetics when he worked with pea pods and pea plants and documented inheritance. So he just used the experimental method with quantitative analysis. So that is working with data that's actually countable and measurable, so like numbers and that sort of thing. And it was a very good example of the scientific method. He worked with pea plants. He bred the pea plants to establish populations of true breeding individuals. This means that the true breeding individuals are individuals that are homozygous for a specific quality. So for example, homozygous dominant or homozygous recessive. The most common example is the color of the pea plant, so either white or purple, where purple is the dominant quality and white is the recessive quality. So he worked with either plants that were true breeding for the purple characteristic, which means that they are homozygous dominant, or they were true breeding for the homozygous recessive. So he cross-pollinated these true breeding parents. We always call the first generation parents with a capital P. And then he raised the seeds of these parents and observed the traits in the first filial generation, so the first offspring generation, which we call F1. He then allowed these offspring to cross-pollinate with each other and observed the second generation, F2. So if we look here, if we look at cross-pollination between purple flower and white flower, where we know that they're both homozygous, so homozygous recessive, homozygous dominant. Obviously, the anthers of the purple plant are removed so that it cannot self-pollinate, only cross-pollination occurs. And we see that all purple flowers result. But we know that because it went with homozygous recessive and homozygous dominant, all the offspring are going to be heterozygous. So he collected data for 7 pea traits: flower color, seed color, the shape of the seeds, pod color, whether the pods were inflated or constricted, whether they were axial flowers or terminal flowers, and the height of the plants. And this was the data he collected about the F2 generation. The most common example is purple flowers and white flowers. So looking at the true breeding purple flowers, as I said, so that's homozygous dominant, and true breeding white flowers, that's homozygous recessive. We want to label, we know that we're going to work with Punnett squares or genetic crosses. So we need to give letters that represent the characteristics. We know that the dominant trait will always have a capital letter and the recessive trait will have a lowercase letter. But we don't want to work with P because capital P and lowercase p are very similar. They could be confused. So we're going to work with F. Obviously, you can choose any letter you like, but I chose F because F for flower. So we know that the dominant characteristic, which would be purple, gets a capital letter, so a capital F, and the recessive generation, the recessive characteristic, which is white, gets a lowercase letter. We know that both these plants are true breeding, so they must be homozygous for their characteristics, which means it gets capital F, capital F. The true breeding white flower gets lowercase f, lowercase f. Now we crossbreed these two species, these two plants. So we can see here we've done a Punnett square, where there's a purple plant, homozygous dominant, and the white plant, homozygous recessive. And all 4 or 100% of the offspring are heterozygous. Because they're heterozygous, they have the gene or the allele for the purple flower and the allele for the white flower. However, since the purple flower is dominant, this is the trait that's expressed. So they're all going to be purple flowered plants. That's what we see in the F1, and we write it as 100% of the plants are heterozygous, so capital F, lowercase f, and 100% of the plants are purple flowered. These plants then cross-pollinate within themselves, and they got to produce the next generation. Since we now have 2 heterozygous plants breeding together, we have capital F, lowercase f, and capital F, lowercase f. When these interbreed, we get F, F, we get 2 capital F, so that's homozygous dominant. We get capital F with lowercase f, heterozygous, and the same outcome here. So two heterozygous outcomes, and we get lowercase f, lowercase f. That's two lowercase f, so that's homozygous recessive. So now we can see that out of the 4 offspring, 1 in 4 is homozygous dominant, 1 in 4 is homozygous recessive, 2 in 4 are homozygous, are heterozygous. So it's 25%, 25%, 50%. So that would be the genotype ratio. If we look at the phenotype ratio, the phenotype is the physical characteristics, so the actual displayed characteristics that we can see. So when we're looking at the plant, when we look at this plant, we don't know immediately if it's heterozygous or homozygous dominant. We just know it's purple. That's what we're focusing on for the phenotype ratio. So we're just going to compare whether they're purple or whether they're white. And out of these offspring, 3 are purple and 1 are white. So 3 and 4 are purple, 1 and 4 is white, which means that it's 75% to 25%, or you can write it in the ratio 3 to 1. As I said, in the genotype ratio, it's 1 to 2 to 1, with homozygous dominant to heterozygous to homozygous recessive. What did Mendel's findings mean? He showed that some traits come in alternative versions, so like purple or white flowers. Those are 2 different versions. These different versions are called alleles. They're the same genes, but they display slightly different physical characteristics. So the different alleles vary in the sequence of nucleotides at the specific locus of a gene. So we know that alleles code for proteins because they're genes. So different alleles which have different nucleotides cause there to be different amino acids brought by the tRNA molecules, which we discussed in the RNA video. Because there's different amino acids brought, there might be a different protein form, and therefore the different alleles will cause different outward characteristics, because there might be the protein that causes it to look like a white flower or one that causes it to look like a purple flower. The traits are inherited as discrete units, so they're not inherited all together, is what it's saying. It's for each characteristic, an organism inherits 2 alleles, 1 from each parent. So there's 2 separate discrete units, 1 from the mother and 1 from the father. This is why it's a diploid organism. We know that diploid organisms have, in humans, 46 chromosomes, so 2 N chromosomes, and all body cells are diploid organisms. Gametes are haploid. They're homologous chromosomes, and it's basically like having 2 different editions of the encyclopedia. It does the same job, but with slightly different effects. One of the advantages of being diploid is that the chromosomes can act as like a backup for each other, so that if one gets damaged or one gets a mutation, the other homolog can silence it and take over almost, like this little cartoon here. Mendel's findings also showed that some traits mask other traits. So one trait is dominant and one trait is recessive. He showed that the traits don't blend. So if you have like a black squirrel and a white squirrel, they're not going to produce a gray squirrel. They're going to produce either a white squirrel or a black squirrel, the chances of each one depending on which trait is dominant. So Mendel saw that purple flowers masked white flowers, which means that purple flowers are the dominant allele. They're always expressed if they're present. The recessive allele has no noticeable effect. The gene makes a non-functional protein. The only time that the recessive allele is expressed is if there's no dominant allele. So if we look at genotype versus phenotype, which I talked about a bit earlier, the phenotype is the description of the outcome of an organism's traits, so the physical characteristics. The genotype is the description of an organism's genetic makeup, so what it looks like inside. So if you try to explain Mendel's results using dominant and recessive, we can say that Mendel discovered that the purple trait is dominant while the trait for white flowers is recessive. He discovered that the phenotype would always be purple if there was a purple gene or an allele for the purple flower involved. The phenotype would only be white if there was 2 white alleles. And the genotype, if the genotype was homozygous dominant, so capital F, or if the genotype was heterozygous, so capital F, lowercase f, then the phenotype would be purple. if the genotype was homozygous recessive, so lowercase f, then the phenotype would be white. We do genetic diagrams using representative letters. So in this presentation, they've used capital P and lowercase p, which is fine for in text because when we're typing, we can see the difference between the capital P and the lowercase p. But when you're writing it out, it's better to use more definitive letters where the capital letter and the lowercase are more defined, more different, because otherwise your marker might get confused and think you don't really understand what you're talking about. So in this presentation, they used the dominant, so the allele for purple, as capital P, and the allele for white as lowercase p. So once they ask you in the test or in the exam, what is the genotype? Don't say homozygous or heterozygous or whatever it is. Say capital P, or lowercase p, or capital lowercase. So give the letters. If you just look at an example of a Punnett square, there's a few rules, or just like things that will help your knowledge. If you cross a true breeding homozygous dominant, so a homozygous dominant with a homozygous recessive, you'll always get a heterozygous offspring. So there's a 100% chance of the offspring being heterozygous, and obviously they'll show the dominant trait. This is called the law of dominance. So the phenotype ratio is 4 to 0, from purple to 0, and the genotype ratio is 0 to 4 to 0, of capital P to capital P, lowercase p, to lowercase p. So if you want an offspring which has a chance to express the recessive gene, both parents has to have a recessive allele in it. As you can see here, this parent doesn't have a recessive allele, so the offspring cannot express the recessive gene. The second rule is that if you cross two heterozygous parents, as you can see, this is why it's hard to use letters such as P. This is a capital P and this is a lowercase p. So these are both heterozygous parents. The rule is that if you cross two heterozygous parents, you'll have a 3 to 1 phenotype ratio of dominant to recessive, i.e. of purple to white. So there's a 25% chance of getting a homozygous recessive offspring. But your genotype ratio is 1 to 2 to 1. When you're stating the genotype and the phenotype ratios, don't just state 1 to 2 to 1. State genotype ratio, capital P to capital P, lowercase p to lowercase p, and then state 1 to 2 to 1, so that your marker knows what you're talking about. You're not just writing down random numbers. These numbers have a meaning. The convention is that when you write a Punnett square, you put the male at the top and the female at the side. This isn't always, like, it isn't vital. You aren't going to lose marks for not doing it, but it's obviously good to have it, especially if they do tell you which is female and male. And because of this, you can often see which is the female and which is the male if they give you a Punnett square. So obviously, just a note that this video is talking about Punnett squares. There's also another way to do it using genetic crosses, I think, but I didn't learn that way. So I'm going to focus purely on Punnett squares. You can have a look at this Punnett square where I think it's for eyes, where brown eyes is a dominant trait and blue eyes is the recessive trait. And these are the different parent combinations. So you can just have a look, maybe try and do it yourself and make sure you understand. If we look closer at Mendel's work, we know that we started with two parents which are true breeding, one true breeding dominant, one true breeding recessive. They gave 100% purple flowering plants, so 100% heterozygous. The offspring of that, so the second generation, gave 75% purple flowering plants with 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. This is another example of how you'd write out the Punnett square for that. The terminology, I'm sure by now you know, but if you don't, homozygous means that the alleles are the same. And for homozygous, you need to specify homozygous dominant or homozygous recessive, because it can be dominant or recessive. Both of them have the same alleles. For heterozygous, it has different alleles, and we know that for heterozygous, there's only one possible way of being heterozygous, and it will always display the dominant trait. So two organisms can have the same phenotype but different genotypes. That would be homozygous dominant and heterozygous, where you can see they're both purple flowers, but one has capital P, one has capital P, lowercase p. When you're looking at a phenotype that displays the dominant trait, so in this case purple, we can't tell immediately if it's homozygous dominant or if it's heterozygous. We can't tell that by looking at it. So then how do we figure it out? What we do is we do a test cross. We crossbreed this individual that we don't know. So we're still trying to figure out if it's homozygous dominant or heterozygous. We crossbreed it with a flower that shows the recessive trait. Because it shows the recessive trait, we know for sure that it's recessive, that it's homozygous recessive. There's no other way, because if it was heterozygous, it would be purple. So we know that this would be lowercase p, lowercase p. We're not sure what this is. If this was homozygous dominant, so if this was capital P, and we cross them, we know from the Mendel's law of dominance that the offspring would be 100% heterozygous. So the offspring would be 100% purple. If this was heterozygous, so capital P, lowercase p, and we crossbreeded them, we'd have 50% of the offspring that are homozygous dominant, 50% of the offspring that are homozygous recessive, so lowercase p, and 50% of the offspring that are heterozygous, which means that 50% of the offspring are white and the other 50% are purple. So what we do then is we continually crossbreed these two species, these two plants. If at any point they produce a white offspring, we know without a doubt that this must be heterozygous. Because if this was homozygous dominant, they would never produce a white offspring. They would only produce heterozygous offspring, which are fully purple. So if we are crossbreeding them and they're continually producing purple, and we do it multiple times over and over, and they never produce a white individual, we know that chances are almost 100% sure it's homozygous dominant. Obviously, we can't be ever 100% guaranteed sure, because it could just be that probability is going a bit crazy, and by chance they're never actually getting a recessive individual. But don't let that confuse you. Just know that if you crossbreed them and they're continually producing purple, this plant must be homozygous dominant. Just know that if at any point they produce an offspring with the recessive characteristics, so in this case a white plant, then this plant, the unknown plant, must be heterozygous. As you can see, these are the two possible outcomes, where it's 100% heterozygous or 50% heterozygous and homozygous recessive. If we look at Mendel's laws of heredity, the first law is the law of segregation. This is slightly different to the term random segregation, which we spoke about during the meiosis video. What this basically says is that when the gametes are produced during meiosis, homologous chromosomes, so the chromosomes where one's from the mother and one's from the father, they separate from each other, and each allele for the trait is packaged into a separate gamete. So basically what that's saying is, if we look at the trait we've been looking at this whole video, the trait for petal color or for flower color, in the parent plant, it obviously has 2 chromosomes and 2 alleles for flower color. What the law of segregation says is that when it produces gametes from the original parent plant, when this parent cell splits during meiosis 1 into 2 haploid cells, the 2 alleles for flower color will each go into separate daughter cells. So what it's saying is that there's not going to be that one daughter cell suddenly gets both alleles for the petal color and one daughter cell gets no alleles. Always each daughter cell gets one of the alleles from the homologous pair. We can see that the meiotic event that creates this is meiosis 1, because this is when homologous chromosomes separate. If we then look at monohybrid crosses, this is what we've been looking at the whole time. This is when it's the inheritance of single characters. So it would only be capital P, and lowercase p, lowercase p. It wouldn't be multiple alleles that cause this trait. There's only one allele causing the trait or in control of the trait. If we just look at dihybrid crosses, some of Mendel's experiments follow the inheritance of two different characteristics, for example, seed color and seed shape, and these are dihybrid crosses. What that's saying is that he chose plants with two characteristics. So they were true breeding yellow round peas, which meant that they were true breeding for the yellow color, and they were true breeding for the shape, the roundness, where the yellow color and the round peas are both the dominant characteristics. So it's homozygous dominant for both color and shape, whereas this pea is true breeding for green and wrinkled. So it's homozygous recessive for both color and shape. He then crossbreeded them, and he only got yellow round peas. We know this because they are heterozygous for both traits. So they're capital Y, lowercase y, heterozygous for color, and because they're heterozygous, they'll show the dominant characteristic, which is the yellow color. They're also heterozygous for shape, which means they'll show the dominant characteristic, the round shape. These then cross-pollinate or self-pollinate either way. And in the final second generation, there's 9 out of 16 yellow round peas. So we don't know, these can either be capital Y, capital R, or capital Y, lowercase y, capital R, lowercase r, or these can be capital Y, capital R, lowercase r. So they can be homozygous dominant for the yellowness and and heterozygous for the round peas, as long as we know that 9 out of 16 of them show the characteristics of homozygous dominant for both characteristics. 3 out of 16 are green round peas, so they're dominant for the shape, the roundness, and they're recessive for the color. 3 out of 16 are yellow wrinkled, which means they're dominant for the color, recessive for the shape. And 1 out of 16 are green and wrinkled, so 1 out of 16 is recessive for both. And obviously, just by looking at this, we can't tell the exact genotype because it could be a whole range of options. Now, they're not going to ask you to draw a Punnett square on dihybrid crosses. It's a lot more complicated. But basically, what you've got to understand is that some traits can come in, what this is trying to show is that one trait doesn't affect the other trait. But just know what a dihybrid cross is. So Mendel is saying, are the alleles for seed shape and seed color handed out separately or handed out together? So when a plant gets, or when a daughter cell gets the alleles, can it only get dominant for both and recessive for both, or can it get dominant for one, recessive for the other? We're saying it can get this because we see here that this can be dominant for one, recessive for the other, dominant for one, recessive for the other. That's what Mendel's trying to show. And this is the second law of heredity, the law of independent assortment. This just states that each of a pair of alleles segregates into gametes independently. So the fact that a daughter cell has the dominant allele for one trait, for example, seed color, doesn't affect whether it gets the dominant allele for another trait, for example, seed shape. So the fact that the daughter cell has the allele for yellow seeds doesn't affect whether it gets the allele for round seeds or wrinkled seeds. That's all Mendel's law is saying, and that's all these dihybrid crosses are showing you. But the only exception to this rule is that if the alleles are on the same genes, on the same chromosomes, because obviously it's not going to get half a chromosome and the other half a chromosome is going to go into another gamete. If, for example, eye color and hair color are on the same chromosome, then the daughter cells will get both and they will affect one or the other. for example, if this was a human cell, we can see that these are a homologous pair, and this is a homologous pair. This is the cell before any meiosis has occurred, and these are the 4 daughter cells. So this basically just skips meiosis 1, the intermediate stage. But we can see that if, for example, hair color was located, hair color and eye color were located on the same allele, on the same chromosome. So each of these chromosomes both have hair color and eye color. Let's look at this black one, for example. If, for example, this chromosome had the allele for blue eyes and the allele for blonde hair, then this cell is going to have the allele for blue eyes and the allele for blonde hair. This cell will also have the allele for blue eyes and for blonde hair, no matter what. So it can't be that this cell has the allele for blue eyes and this one has the allele for blonde hair. Those 2 alleles can't be split up because the alleles are located on the same chromosome. But that's just the exception to the rule. The main part of the rule, or the most important part that you need to know about this law of independent assortment, is that in general, each of a pair of alleles segregates into gametes independently of another pair of alleles. So the allele that a cell receives for one trait doesn't affect the allele that a cell receives for another trait. That's basically what this is saying. And meiosis 1 also creates the law of independent assortment. You can just look through this and you can understand how the meiosis and gamete formation and fertilization lead to the variation in the offspring. So if we just look over Mendel's laws of heredity again, we know that the law of segregation relates to monohybrid crosses for single traits, and the law of segregation just states that each allele segregates into separate gametes. So the maternal and the paternal alleles segregate into separate gametes. They'll never go into the same gamete. Then the second law, the law of independent assortment, relates to dihybrid crosses, where there's 2 or more traits, and this states that each pair of alleles for genes on separate chromosomes segregate into gametes independently. Mendel's choice of P's was wise. They're good for genetic research because there's a whole lot of different varieties with distinct heritable features. So distinct, we can tell the difference. For example, purple and white. It's easy to tell the difference between a purple plant and a white plant, and they're heritable features, so they're passed on to different generations. Mendel had strict control over which plants mated with which. Each pea plant had male and female structures, so Mendel knew that the plants could self-fertilize, but he could also cross-pollinate the plants by moving the pollen from one plant to another by himself. Pea plants are quite simple genetically, most characteristics controlled by a single gene, which made Mendel's work a lot better for genetic research. And each gene has only 2 alleles, one of which is completely dominant over the other. Also, pea plants are small, so you can keep a lot of them. They're not expensive, and they produce a lot of offspring, which obviously is important because this makes the sample large enough to reach valid conclusions. If the pea plants only produce like 3 offspring in their whole life, that's not enough for Mendel to say, okay, well, to draw enough valid conclusions. And we know this from experiments that you have to repeat the experiments to make the conclusions reliable and valid. Pea plants are also a good plant to use because it doesn't take too long to obtain the next generation.